Welcome to part 3 of this video lecture. The topic of this part is validation and quality control, which, as you will hear, are related but not the same. Both validation and quality control are done to demonstrate that the results of an analytical method are reliable, or perhaps I should say reliable enough for what they are used for. Very generally, we want to ensure that the result that is reported is an accurate reflection of what it was at the time of sampling. Method validation therefore focuses on accuracy, precision, selectivity, and stability. The first question is, when is a result good enough? This depends quite strongly on the application. Sometimes a rough estimate is sufficient, and sometimes it is really important that we accurately determine the concentration. But in practice, there is a strong desire to always use the same criteria, at least for bioanalysis that supports drug development by the pharmaceutical industry. And therefore, most bioanalysts use the criteria that have been issued by regulatory authorities in a number of international scientific guidelines. The most important are the guidelines of the US Food and Drug Administration, or FDA, and the European Medicines Agency, or EMA. But various other guidelines also exist. Let's have a look at what these validation guidelines say. First of all, we distinguish full validation and partial validation. Full validation is performed for each new method and for a new analyte or a new matrix in an existing method. It is an extensive assessment of all important characteristics of a method. Partial validation is done when an already validated method is slightly adapted. For example, when an extraction method has been changed. It's a more limited investigation of only the aspects that are affected by the changes in the method. Cross-validation is something else. It is a comparison of different methods in one lab, or of one method in different labs, and it simply is the analysis of a single set of samples by the different methods or different labs. Now the most important validation parameters. Precision. It expresses the variability between results when one sample is analyzed multiple times by the same method. Accuracy describes the difference between a measured result and the true or the theoretical value. This can be visualized with a dartboard. If the true value is represented by the bullseye, and we analyze or throw 4 times in this case, a measurement is accurate when the mean of the 4 throws is exactly at the target. A measurement is precise if all 4 darts hit the same place. And of course, ideally a method gives both accurate and precise results, which corresponds to throwing all darts into a bullseye. Precision can be subdivided. Repeatability, or within-day precision, is the possibility to properly repeat a measurement under identical conditions. And reproducibility, or between-day precision, is the potential to have a result reproduced by the same method, but under conditions, other conditions such as another analyst or other materials. As an example, let's have a look at the method for risperidone in human plasma by LCMS. A sample was prepared at 0.1 nanogram per milliliter and analyzed 10 times, which gave 10 different results. From these results, an average and a standard deviation can be calculated, and a coefficient of variation, or CV, and a bias can be determined, and the bias is then the deviation of the average result from the theoretical value. The CV is used as a measurement for the precision, and the bias as a measurement for the accuracy. With sensitivity, many people refer to the potential of a method to determine very low concentrations, but its actual meaning is the ability to detect small changes in analyte concentration. Steep calibration curves mean a high sensitivity. In this ELISA curve, the steepest middle part represents the most sensitive concentration range. In the outer parts, the same change in concentration give much less difference in response. So these are the insensitive parts of the curve. The quantification limits of a method are the lowest and highest concentrations that can still be determined with a suitable accuracy and precision. These are called the LLOQ and ULOQ, and they are typically based on what is expected and relevant in a study sample. The interval between LLOQ and ULOQ is called the concentration range of a method. And ideally, there is a linear relationship between concentration and response within this concentration range. For LC-MS methods, this very often is the case. This is shown here for risperidone over the range of 0.1 to 250 nanograms per milliliter. Some more definitions. Specificity is the ability of a method to generate response only for the analyte and no response for any other component in the sample. Very few methods are really specific. One example is the pregnancy test, which only responds to the pregnancy hormone HCG in urine and not to any other urine compound. If a method is selective, it is able to quantify an analyte reliably in a complex sample. Other sample components may also give a response, but a selective method is capable of sufficiently separating these responses, for example by a chromatographic separation. If a compound other than the analyte contributes to the total response, this is called an interference. In bioanalysis, interferences can originate from the matrix, such as endogenous plasma components, but also from drug metabolites or from the materials that are used for the analysis, such as solvents or buffers. For risperidone in plasma, we see that there are some very small responses in the chromatogram of blank plasma, but these do not really interfere with the peak for risperidone. Therefore, this method is sufficiently selective. In another method, the endogenous compound citidine has the same mass and almost the same structure as the drug cytarabine. And if they are not sufficiently separated, citidine interferes with the measurement of cytarabine. The left-hand chromatogram shows insufficient separation between the two, and this method is not selective. The chromatogram on the right shows a good selectivity because both peaks are well separated. Sometimes small changes in the experimental conditions are introduced on purpose to see if a method is insensitive. If not, a method is called robust. Very important is the stability of an analyte during all phases of the analytical process: sampling, storage, and analysis. During validation, samples are subjected to various relevant conditions to demonstrate that the analyte is sufficiently stable. This can be storage at room temperature, in the freezer, and over different freeze-thaw cycles. A nice illustration is what happens to the drug bendamustine in urine when it is stored at room temperature at a few different physiological pH values of urine. As you can see, at a relatively high pH of 8.5, the drug is degraded very rapidly within just a few hours. At lower pH values, this is less pronounced, but to guarantee complete stability, urine needs to be acidified to about pH 2 as soon as possible after it is produced. And finally, extraction recovery. This is the percentage of the analyte molecules that is transferred from the sample to an extract. For example, if plasma is extracted with an organic solvent, part of the analyte molecules are transported to the solvent and the rest stays in the plasma sample. As you can see in this example, 80% of the black circles are recovered in the organic solvent and just 25% of the green diamonds. Now let's have a look at how validation is done in practice. First, calibration and validation samples are prepared. This is done by adding small volumes of a stock solution or standard solution to a blank matrix. It is important that the stock solvent is miscible with water to allow homogeneous distribution of the analytes across the sample. Normally there are around 8 to 10 calibration samples with different concentrations and 4 validation samples. When the validation samples are analyzed and their concentrations compared to the calibration curve, their deviation from the theoretical value should be below 15% or below 20% at the LLOQ level. The precision of the repeated analysis has the same criterion. CV should be below 15 and below 20 at the LLOQ. For samples that have been subjected to stability assessment, the bias also has to be below 15% from the theoretical concentrations. After completion of a validation, a method can be used to analyze clinical samples. During this application of a method, quality control samples at 3 levels are included in duplicate. The method is considered reliable if at least 4 out of these 6 QC results have a bias below 15% from the theoretical or true value, and at each of the 3 concentrations this has to be at least 1. If this is the case, we accept all results of the study samples in this run. If not, they have to be rejected and the analysis has to be repeated. A nice way to visualize the QC results is by plotting them in a so-called Shewhart chart. In this figure, the acceptance limits of 15% below and above the theoretical value are indicated with red lines. And one can now immediately see if a QC result passes or fails, and also trends can be picked up. To conclude, validation and quality control are essential for a reliable analysis of clinical samples. Validation is performed before using the method and quality control during its use. Only sufficiently accurate, precise and selective methods give good results, and in addition, analytes need to be kept stable during all phases of the analytical process. I hope to see you back in part 4 when we will discuss an example of quantitative bioanalysis.